All right, so this is part 2 of chapter 8. We're going to talk about the neural integration and some neurotransmitters in this section. So let's go ahead and get started. So we're going to take a look at some concepts here with divergence and convergence, summation, frequency coding, the axon hillock, and threshold. So just a quick word on convergence and divergence. So when neurons communicate with other neurons, they can do it in, there's different pathways and patterns of those pathways. One general pathway is called a divergent pathway or divergence, and it's basically what you see here, where one neuron has several collaterals. So we can have, remember, collaterals are the branching of axons. So they can have several collaterals, and that way they can communicate with many neurons, many other neurons. And so we can have one action potential spread out and have many downstream effects, incorporating many or different areas of the brain, for example. Convergence is the opposite, where a neuron typically will receive messages from many different neurons, and so they're converging on that one neuron. And when this happens, we can think about the summation process that has to take place, right? So all these graded potentials are either excitatory or inhibitory, and so that neuron that is being converged upon will have to summate those graded potentials. And so there's some neural integration. So this is called neural integration, when one neuron has to integrate the information coming from different sources. And we've looked at this before as well. So this is summation. So you can either, and this is for graded potentials, right? So you can summate graded potentials either temporally or spatially. So the temporal summation here, just a refresher, right, where you have the same synapse. So in this example here, we have AAAA. So we're just looking at this synapse, which is an excitatory synapse. And you can get a depolarization, sub-threshold depolarization here. A little bit later, you have a sub-threshold depolarization again. So when they are spread out time-wise, each of those are not going to create an action potential. However, if they come close together, right, if the timing is such that they are in quick succession, then the additive effects of both of those smaller depolarizations add up. So they summate, right, until they actually do reach threshold potential, and then an action potential is produced. So temporal summation just means that timing, right, if you have very quick action potentials coming at one time, right, so this is that whole idea of frequency coding. If you have a lot of action potentials coming very frequently, one right after the other, you can have a summation happen, where you can, it's more likely that you get an action potential coming on the postsynaptic end. Spatial is where you can have many synapses. So now we're looking at all the synapses, A, B, and C. A and B are depolarizing, they're excitatory, and then C is inhibitory, it's hyperpolarizing, right? But you can summate. So if A, B, and C fire separately, they each of them alone are not enough to cause an action potential to be generated. But if A and B occur together, right, if you spatially, if they both depolarize, or if you get the binding of the receptors together, then you can summate that and you can reach threshold potential and have an action potential. There's also summation with the hyperpolarization, right? So if you have A and C together, right, so A is a slight depolarization, C is a slight hyperpolarization. Together, you get basically the resting potential. So this is, you're not going to get an action potential, but this is also inhibitory. All right, so that's just a review of the two kinds of summation patterns that we see with neural integration. And then also just reminding you, we talked about this before with frequency coding. So the degree of depolarization at the axon hillock is signaled by the frequency of action potentials, so frequency of action potentials. So when you summate, right, that can affect depolarization. Summation therefore influences the frequency of action potentials. So if you have more action potentials result in more neurotransmitter released, and consequently a greater inhibitory postsynaptic potential or excitatory postsynaptic potential in the next neuron. So basically what this is saying is the more frequent action potentials come, then they're going to result in more neurotransmitter released into the cleft. And if there's more neurotransmitter present, then you're going to get a greater graded potential, right? Because you have more intense signal with neurotransmitters. You get more binding. And so you can either have a greater, if it's an inhibitory signal, a greater inhibitory postsynaptic potential, or you can have a greater excitatory postsynaptic potential. So the frequency of action potentials code how intense the signal is going to be. All right, so let's talk about the next topic here, which is the presynaptic modulation. So before we have the synapse, we can actually modulate or control or somehow fine-tune, either turn up the volume or turn down the volume of the active potential. So the regulation, this is a regulation of communication across the synapse, and this is an axonic synapse, so it's between two axons, and they're going to function as modulatory synapses. So they're not going to stop an action potential or create an action potential, but they're going to modulate the action potential. So there's something called presynaptic facilitation or presynaptic inhibition. And so just looking at the terms here, here's our presynaptic neuron, right? So the action potential is coming down this way on the axon. Here's our postsynaptic neuron, so this is receiving the signal, right? This is on the receiving end. And then here's our modulating neuron. So the neuron can actually come in and before the synapse, right? That's why it's called presynaptic facilitation or presynaptic inhibition. Before the synapse, you have some communication right here, and that's going to modulate what's going to happen at the synapse. So this is just a quick look at presynaptic modulation. This is going to be facilitation. So facilitation, that word means when you facilitate something, you make it happen easier, right? You make like a facilitated diffusion, you have a transport protein that's going to make it easier for facilitation to happen. So this is going to be an increase of neurotransmitters. So you can see, or an increase in the excitatory signal. So here's our action potential. Our action potential is coming down. So we can see how E here is going to be a modulator of C. So E is going to also send an action potential down. And you can see that when E is not firing an action potential in our first case here, when it's not firing, we have a certain amount of neurotransmitter that's being released. But the action potential coming from E, if this is going to be a presynaptic facilitation event, then we're going to communicate, E is going to communicate with C is going to create a lot more neurotransmitter, or the exocytosis of neurotransmitter is going to be much, much greater. So we're going to have an enhanced excitatory or inhibitory signal, but you're going to modulate with a greater amount of neurotransmitter. Okay, so this is just taking a look at, so we have C alone, D alone, E, right? E is not actually contacting X, the neuron here. But with C and E, right, you're going to have an increased, you're going to create an action potential and increase the amount of neurotransmitter being created. Let's look at our next image here. So this is called presynaptic inhibition, and it's pretty straightforward. So you can see that H is going to inhibit F. So when an action potential is coming down F and H is silent, you get a large amount of neurotransmitter being created. But if H is also firing with F, then H is going to somehow inhibit, right, or turn down the volume on how much neurotransmitter is going to be secreted. So it's going to inhibit that postsynaptic membrane. All right. So what happens during the presynaptic facilitation? Okay, if you're facilitating, then the answer is A. All right, so now we're going to turn our attention to different kinds of neurotransmitters. And this is a table that neatly put them in, in one picture so you can see what we're going to talk about. So we're going to talk about acetylcholine. So some biogenic amines. These are amines, and they're derived from amino acids, but they're not amino acids. This third category, they actually are amino acids. Then we have some purines. You're familiar with ATP. We have some things called neuropeptides, which are small proteins, right? Peptide means protein. And then some unique molecules that are sort of outside the box. They don't belong into a category. So let's take a look at our first one, which is acetylcholine. So acetylcholine is made inside the cell. It's made in the axon terminal, which is unique because a lot of neurotransmitters are made in the soma, and then they are transported down the axon and stored in the axon. But this one's actually created and synthesized in the axon terminal. And the enzyme that is present to do this is choline acetyltransferase. And so basically it takes the precursor. This should look familiar to you. This is from Krebs cycle. And it puts a choline on to make acetylcholine and coenzyme A. So this is our neurotransmitter. And then when acetylcholine is broken down, the enzyme that does that is called acetylcholine esterase, and it occurs in the synaptic cleft. And what happens is you just break it down again to, well, actually the precursor is not the same. Acetylcholine becomes acetate and choline. So let's just take a look at this picture of the process. So here's our acetyl-CoA and choline. This is driven by the enzyme CAT. We get acetylcholine, which is then stored inside of our vesicles. All right, so when it is exocytosed, the receptors for this acetylcholine are called cholinergic receptors. So I do want you to know the term cholinergic receptor. And neurons that secrete acetylcholine and synthesize acetylcholine are called cholinergic neurons. So this would be a cholinergic neuron. And there's actually two kinds of these cholinergic receptors. There's a nicotinic and muscarinic. We'll take a look at that. And then here is our enzyme. So here's acetylcholine esterase. This is the enzyme that degrades acetylcholine into choline and acetate. And then these substances are taken back up so they can be recycled, right? They can be reused to build another acetylcholine. So anytime you have neurotransmitter in the cleft, remember that there's three fates that can happen to them. One, they're degraded. Two, they can be reuptaken. Or three, which is not pictured here, they can just diffuse away. So let's take a look at the receptors. This is a nicotinic cholinergic receptor. So you can see ACH, that stands for acetylcholine, binds to the receptor. And it's actually, there's two binding sites. And then it's basically an ion channel, right? So this, what kind of channel or action is this? This is an ionotropic, right? So this is a very fast receptor. It's going to give you a fast EPSP, right? So excitatory postsynaptic potential. You'll find, if you have studied this in anatomy, that muscle contraction, skeletal muscle contraction, is based on acetylcholine neurotransmitter. And these are the kind of neurotransmitters you find on skeletal muscle cells, the nicotinic cholinergic receptors. Then you see here another kind, muscarinic cholinergic receptor, which is a G protein coupled receptor. So when you see that, you think, okay, this is a slow kind of receptor, right? It's going to ionic receptors, ionotropic receptors are fast. If you have a G protein coupled receptor, it's going to be slow. So here's our muscarinic cholinergic receptor. Here's our binding of acetylcholine. We have a lot of proteins associated with this receptor, and we can see we can have direct activation of an ion channel, or we can have activation of a secondary messenger system. So they're both something that are possible. And just so you guys know, muscarine is something that can be found in poisonous mushrooms, the mushrooms that can kill you. And it is used for medicine for some sorts of problems. All right. So to which receptors does acetylcholine bind, and what are the resulting effects? All right, so we're going to look through this. So nicotinic cholinergic metabotropic, muscarinic, cholinergic, ionotropic. That's incorrect, right? Nicotinic cholinergic is ionotropic, EPSPs, muscarinic cholinergic is metabotropic. That sounds right. Nicotinic cholinergic, ionotropic, IP, nope, right? It's not inhibitory. And nicotinic cholinergic, ionotropic, EPSPs, muscarinic cholinergic, ionotropic, no. All right, so answer B is our correct answer. All right, so our biogenic amines, all right, so these are going to be neurotransmitter that are derived from amino acids. So you need amino acids in your diet to make these, but they are not amino acids themselves. So there's a class called catecholamines, which are derived from an amino acid called tyrosine. And you've probably heard of some of these before, dopamine, norepinephrine, and epinephrine. You've probably also heard of serotonin and histamine. So histamine is also secreted by your immune system that causes the inflammation and itchiness and swelling of allergy. But it also functions as a neurotransmitter. So when you have these neurotransmitters produced, they are released into the cleft, and they're going to also be degraded, and there are enzymes for degradation called monoamine oxidase, and then this COMT. I'm just going to emphasize monoamine oxidases, or MAOs, because there's a class of drugs called MAOI, which is here, monoamine oxidase inhibitors, which are responsible. So if you're breaking down your MAO neurotransmitters, right? If you're breaking down your serotonin, dopamine, and norepinephrine, epinephrine, for depression disorders, right? They think that the action of dopamine, norepinephrine, and serotonin is not adequate in the brain. So either you're not making enough of it, or there's just not enough activity in the cleft. And so these inhibitors want to increase the levels of the action of these neurotransmitters by stopping the degradation, right? So you want to inhibit the enzyme that breaks down these neurotransmitters so that the neurotransmitters can be more active in the cleft, right? So these inhibitors will increase the concentration of those three neurotransmitters. And when you have more of these neurotransmitters, it can relieve the symptoms associated with depression, sadness, or anxiety. So that's one look at, targeting enzymes inside the CNS to alleviate depression. And the other class of drugs are SSRIs, or selective serotonin reuptake inhibitors. And this is where they target the reuptake mechanism. So they block that protein so that the serotonin, when it's released into the cleft, lasts longer. It's not taken away back up into the presynaptic membrane. So those are two classes of antidepressants. And just to be aware that both of these have a lot of different side effects. Because these neurotransmitters have wide-ranging action in the CNS, and so they can have wide-ranging action in the body. But I want to point out that you can read about this in page 210 in your book. There's a clinical little paragraph there, or section. All right, moving on to adrenergic. The adrenergic receptors are the receptors for the epinephrine and norepinephrine. So adrenergic, the adrenaline, right, the adrenal glands make epinephrine and norepinephrine. So adrenergic, it refers to the adrenals. So these are going to bind to catecholamines, specifically norepinephrine and epinephrine. And so we can see, right, we can think about adrenaline, and adrenaline, when you release it into your body, the adrenaline sets up your fight or flight response, your sympathetic autonomic response. So you're going to have an increased awareness, your heart rate's going to go up, you're going to be ready to fight or flight. And so you can see that a lot of the target cells that have this adrenergic receptor are going to be parts of the body, your heart, your blood vessels, your airways of the lungs, your iris dilator muscles. So they're going to all have an effect on these smooth muscles of the body so that you can increase the breathing rate, you can increase the heart rate, you can dilate your pupils so you let more light in, so you can sort of see better in dim instances. But this is all setting you up for fight or flight, or the sympathetic nervous system response. The next topic is going to be the amino acid neurotransmitters. And so the two that I want you to keep in mind are glutamate and GABA. So glutamate is going to be predominant excitatory neurotransmitter in the CNS, and GABA is going to be your predominant inhibitory neurotransmitter in the CNS. And so it's just, these are amino acids just straight up from protein acting as neurotransmitters. And, I don't have another picture, but there is a blurb again in your textbook about the role of GABA. All right, so GABA is this nice long word here. So because it's an inhibitory neurotransmitter, it quiets down the activity of the central nervous system. So people who have anxiety or maybe PTSD or some kind of hyperactive, if you're feeling, anxious or very, very nervous, you can take, medicines like Valiums or sedatives that basically they work on the GABA receptors. So if you look at the outline, I do mention that agents that increase GABA activity will suppress the central nervous system, things like sedatives, things like benzodiazepines at low doses, and even alcohol, which will enhance the effect of GABA, which then will quiet or inhibit the central nervous system. Of course, too much alcohol or Valium or anything can kill you, right? Because you can depress the central nervous system so much that it stops working, and then you have a problem. And also what's dangerous is taking these drugs together, because they all bind to the same receptors, and so they will have a summation effect, right? They're going to have a synergistic effect, and they're going to have an even more profound effect on inhibiting the central nervous system. So like drinking wine and taking benzodiazepines, that's a very dangerous mix. The next neurotransmitter are purines. So ATP is something that you're very familiar with as an energy source of cells, but it also can act as a neurotransmitter. And a lot of the receptors for ATP as a neurotransmitter are in the gastrointestinal tract, but also in the CNS and PNS. And it's broken down just like any other neurotransmitter is in the cleft by nucleotidases or nucleotidases, right? The nucleotide ACE. But this is just kind of an example of some what ATP, you should know what ATP looks like, and then ADP, and then GTP can also be purine neurotransmitter. All right, moving on to neuropeptides. So neuropeptides are short chains of amino acids. Most of them are co-localized with other neurotransmitters. That means that they are together with the other neurotransmitters, and these guys can modulate responses by other transmitters. So if you look through the list, some of these you may have learned before as hormones. And so classically, many of these are called hormones. So there's thyroid releasing hormone, there's antidiuretic hormone, there's oxytocin, which is in the hormone endocrine system. But then there's substance P, which I don't know if anybody's heard of this, but this can decrease your gastrointestinal motility. And then there's something called CCK, which is not on this slide, but I have it in the outline. Cholecystokinin, which is also in the gastrointestinal tract. It helps to release bile from the gallbladder. And then there's a new one called orexin, which is involved in sleep-wake cycles. But then there are these endogenous opioids. Okay, so we have neurotransmitters called endogenous opioids. So they're called enkephalins and endorphins. And you've probably heard of the word endorphin before because, runner's high, or, when you exercise, you do physical activity, or something really, really good happens to you, you have these natural endorphins in your brain that are released and you feel really good. So they're involved in the reward pathway, and they make you feel happy and good and, high on life. And also a role of these opioids is these endorphins and enkephalins is that they can modulate pain. So the more of these endorphins and enkephalins, your pain perception goes down, which is why a lot of your pain medications for severe and chronic pain act on these opioid receptors. So if you're giving a person an opioid drug, it's going to activate these pathways, and the pathways, will mitigate pain, which is the primary job of these opioid painkillers. But the secondary effect is that, they do produce a high and they make you feel good, and that's why they're super, super addictive. So anyways, this is just a note on that we do have this endogenous opioid system. The opioids that we make fall into these two categories. And, the opioids that you take for drugs like morphine, for example, will act on these same pathways. And then to our unique neurotransmitters, there's two that we're going to mention. So this is nitric oxide and endocannabinoids. So they're unique because they're not stored inside of the cells, that they're at least nitrous oxide, nitric oxide, not nitrous. Nitric oxide is not stored, and it's released immediately once it's made. So there's an enzyme that can create nitric oxide. It's tiny, it's a gas, and it's permeable through membranes. And so once it's made, it diffuses out of the cell, and it travels in the bloodstream, and it can reach its target cell. So it's unique in the fact that it doesn't have to work at the cleft, that it can diffuse out away from that cleft and have action further out. But it doesn't have action way further out because it's actually a pretty quick molecule. It breaks down really, really quickly. And so it can only be active in a very small area, right where it diffuses into. So it breaks down by itself. There's no enzyme that breaks it down because it has a very short lifespan inside your body. And one of the things that nitric oxide does, it's a very good vasodilator. So it relaxes the smooth muscle, right? Your smooth muscle here in your blood vessels, relaxes them, and so it dilates the blood vessels, and you can increase blood flow to certain areas of the body. Or you can also reduce the blood pressure, because blood pressure is that pressure against the walls, and if you open up your blood vessels, the pressure on the walls will go down. Lastly, we have endocannabinoids. So this is a really interesting system. One example of an endocannabinoid we have is called anandamide, and this is made from phospholipids, so it's fatty. And it can also be released, and it binds to receptors on the target cell. And there's a variety of physiological processes that these endocannabinoids are part of. So they can mediate appetite, pain sensation, mood, and memory. And this is an area of a lot of research, and this is a whole system that THC from marijuana acts on. So anandamide is a naturally produced endocannabinoid. THC is from the marijuana plant, right? But they're similar. So that's why THC binds to this endocannabinoid system that we have, and it gives you the physiological effects that THC has. So people are looking at, what the endocannabinoid system, what's the exact function in the body. And there's a lot of research out here. I just thought I'd show you this one research paper from Cell. So Cell is a very prestigious publication that only publishes serious work. So this research is basically looking at metabolism and how the endocannabinoid system mediates obesity and our calorie intake and our metabolism. So you can see that if you wanted to take a look at this, you have up arrow means increased activity by the endocannabinoid system, namely THC, right, from marijuana, decreased, and then the question marks means they're not sure that further research is needed. But just a couple things, right? So one of the funny things I noticed was that you have an increase in fat preference and intake. So maybe eating more fatty foods. Secretion of ghrelin. Ghrelin actually makes you feel hungry. It's a hunger hormone. And then you can see here that the nose, it increases odor sensitivity and it increases food-seeking behavior. So all sort of pointing to that, the sort of munchies sort of thing with people who take THC. Anyways, you can kind of like look through and see what's out there, but you can also see just in general, like look how many parts of your body, right, that have the cannabinoid receptors, CB1 receptors. There's several receptors, CB1, CB2. I just put CB1 in your outline. That's found in the central nervous system, but you can see that the receptors for the endocannabinoids are everywhere. They're in your nervous system, they are in your gastrointestinal system, they're in the liver, they're in the skeletal muscle, and then in your adipose tissue. So it's a widespread system to the body, right? So people are trying to look at like what's the purpose of this, this system in different parts of the body. All right, just wrapping up here with a question: which of these is not a reason nitric oxide works differently from the general pattern of neurotransmitter function? Another way to think of this is which statement is false. So if you read through them, you can pause and think about it, or I'm going to tell you the answer here. It's letter D, right? So it's true that it cannot be synthesized and stored. It's true that it diffuses to the target cells without needing a synapse. It's true that it can enter the cell by diffusion. So it leaves the cell by diffusion, it enters the cell through diffusion. But the only thing that's not true is it cannot travel long distances because, again, it breaks down in seconds. And so it can't go throughout your widespread body wide. It only is localized. And that's the end.